Okay, hello everyone. Myself, Dr. Kushal Salunke, working as a junior resident in Department of Biochemistry, GMERS Medical College, Godri, Vadodara. Okay, we are starting the practical series of biochemistry, and this is the first practical that you must know before you actually went into the deeper part of the other practicals of biochemistry. This is very basic practical. Okay, I hope all of you have started reading the subject biochemistry side by side along with your anatomy and physiology. Okay, so in this practical, we'll discuss very basic things about what are the common instruments that is used in a clinical biochemistry lab. Okay, you all know from our department we have two labs that is working. Okay, you all know. First lab in which you are doing the practical is UG lab. Okay, in which we are performing the qualitative and quantitative analysis. Okay, by the MBBS student, that is you. Okay, that we are performing the practicals at UG levels. And second lab is a clinical chemistry lab in which we are performing different tests by using semi-automated as well as automated analyzer. Okay, that is useful for patient treatment and patient prognosis also. Okay, so in this basic practical we will cover what are the common instruments, why these instruments are important, and what is the role of biochemistry lab, main thing, and what is the role of biochemistry lab, what kind of test we are performing in the biochemistry lab. That all will be discussing in this particular practical. Okay, so let's start the practical. And according to exam point of view, I must say that this practical will cover your two questions in your spotting examination. Okay, so spotting, there is in our university, MS University, will cover 10 mark of spottings in university as well as your prelim examination, and 2 marks for each spot will be there. So approximately you can self 2 to 4 marks will be covered by this particular practical out of 10. Okay, so it is important to note the instruments. You have to identify that particular instruments and you have to mention the use of that particular instruments. Okay, and little bit more we will be talking about the blood collection system and the other vacuities also in this particular practical. We just highlight it, we have a separate practical for that. Okay, so we will be starting the practical. Okay, these are your biochemistry competencies, like you have to commonly describe the laboratory apparatus and equipment. You have to outline the basic functional principle. Okay, how the instrument is working, the basic principle, not in detail, in a biochemistry and their application. Okay, and you have to observe that particular equipment while it is working. Okay, the techniques in the biochemistry lab. Okay, so they will come, it will cover this all three competencies. Okay, according to competency based medical education. So what are the common instruments that is used in a UG biochem lab as well as CCL lab, that is clinical chemistry lab? So first basic instrument is a balance, then magnetic stirrer, centrifuge, hot air oven, incubator, water bath, all will be discussing in subsequent slides. Desiccator, pH meter, thermometer, various glasswares, okay, including your pipettes and beaker all. Semi-automated analyzer, fully automated analyzer, electrophoresis chamber, chromatography chamber also. Okay, electrophoresis apparatus also. Okay, colorimeter, then photometer, and then spectrophotometer. Okay, so starting with the balances. So you can see in the screen we have two types of balances. First image is of electronic balance and second one is a mechanical balance. Okay, we'll discuss what is the difference between it. Okay, so balance first we have physical balances. It is rather a very crude equipment which is used for measuring the chemicals which are needed for making a qualitative reagents. So whenever you are performing a qualitative analysis, we have a reagent, we are using the reagent to measure different tests and to do a different test. So to make this particular qualitative reagent we will require a balance. Okay, because we have to make a specific amount. Okay, and where larger amount of the chemical to be measured, the physical balance is very important. They have double balances where on one side known weight is kept and on the other side the what chemical you want to measure, that particular chemical has to be put on the side of that particular balances. So the weight which can be measured in this instrument is vary up to 1 milligram to 1,000 gram, that is up to 1 kg you can measure. Okay, so electronic balances, they are more accurate, they are more accurate. Okay, so electronic balances are more accurate in nature. Okay, then are being preferred because of accuracy and ease of operation. Okay, they are single plane balances. Okay, here electronic balance mein, you can see there is a single pen. Okay, this is single pen which operates on the principle of electromagnetic force. You should note this. Okay, and counterbalance is weighed a sample mass. Okay, the electromagnetic this balance is giving a reading according to electromagnetic force. It measures rather a smaller amount compared to a physical balance. Okay, very small amount is measured, like from 1 mg to 100 gram only. So facility of telling in the electronic balances makes the measurement very easy, as because it is the weight of container is adjusted by the machine itself. So when you put the reagent or something in a container, that weight of the container has to be eliminated. So electronic balance will do that automatically. So zero setting is there. You have must seen in the various shops also. Okay. Now second instrument is a magnetic stirrer. You can see this is used through the for the thorough mixing of solutes in a solution and to make a homogeneous mixture. Okay, we are using this particular instrument. An iron capsule is placed in the vessel containing the solution, and then this vessel is placed on the pan of a magnetic stirrer plate, which is built for providing a rotatory magnetic field. Okay, so whenever you have to mix the reagent and you have to make a homogeneous mixture, we are using the magnetic stirrer, which is very much useful as compared to your physical stirrer, glass stirrer. Okay, so moment of iron capsule in a solution on twitching the instrument mixes the solution completely as to provide a homogeneous mixture. So you have to note that this is used for thorough mixing of solutes and to make homogeneous solution. Okay, only this much you should know. Now this is very important instrument, centrifuge, and this instrument is commonly used in a biochemistry lab to separate serum from a plasma. I hope by now you should know the difference between serum and plasma. What is the difference between serum and plasma? You have to answer it in a comment section. Okay, so from the whole part, whenever you want to separate a serum and plasma, and why we are separating, because we have to do a test whenever samples received in our lab, we have to centrifuge the sample. Okay, because we are performing the test on serum or plasma. Okay, we are measuring the biochemical enzymes and the markers in the serum and plasma, so whole blood is not required for us. So we have to take a sample that is only containing a serum, so we have to separate it. So in the centrifuge we are using the for the separation. Okay, it is also used for the separating the sediments in the urine. So centrifuge is based on the principle of centrifugal force. You know from your physics classes, there is a centrifugal force when a rotatory motion is applied. Okay, so from this the solid matters will be separated and liquid suspension is formed. So you can see the centrifuge and how the second photograph is also of centrifuge. You can see this both the photographs. Okay, right? Yes, so now we are moving ahead. Okay, centrifuge, so this whole thing is not required to you, but you should know that centrifuge are so many of types. You can see the benchtop model with refrigeration based on rotator design and based on maximum speed. Okay, one point you should note, the principle of centrifugal machine, we have a relative centrifugal force RCF which is applied over that. Okay, and whatever the speed of that particular instrument is designed, which is calculated according to revolution per minute RPM. Okay, in which whenever you are rotating in very high speed, that is 1 lakh RPM, it is known as ultra centrifuge. Okay, you can see this hot air oven. There are two instruments which are look like this. One is hot air oven and second one is your incubator. We'll see in our next slide. Okay, that is hot air oven and your incubator. So first we'll discuss hot air oven. You should note this, that it is the equipment which produces dry heat, okay, and its internal chamber. And the temperature achieved here is a 250 degrees Celsius. Up to 250 degrees Celsius you can achieve the temperature. Okay. You carefully see this particular part where it is very thick wall and double insulated wall. And you should know that here there is a plain door. Okay. Without any window or glass slide. Okay. Here there is no window or glass slide. Okay, Make a note of it. So this instrument is used for drying of glassware. Whenever we have used that particular instrument for qualitative or quantitative analysis, there is a sticking of reagent in that particular instrument. So when I have to do the another experiment with the same instrument, I have to air dry it. So because of it, we have the hot air oven. Heating the chemicals wherever it is required and dry sterilization of glassware in microbiological experiment, metal equipments and swab, etc. Okay. Now you can see this incubator. Okay. In the incubator, we have the glass window or opening window here to observe the whatever is going inside. Okay. Right. So it is an instrument which is capable of maintaining the desired temperature. What level of desired temperature? You can see 60 to 65, maximally up to 100 degrees Celsius. Mainly it is used for microbiological lab in culture plate incubation. Okay. So in your second year, you are knowing this is a culture plate in which I have performed the streaking or lawn culture of that particular organism from the sample. And if I incubate, I can see the colonies of bacteria, of suspected bacteria after incubation. So this is how the microbiological experiments are actually happening. Okay. So in a biochemistry lab, this incubator is also used for incubate the reaction mixture at specific temperature for a set duration, which depends on the method of adopted assessment. Okay. Whenever we want to perform a one reaction. Okay. For suppose estimation of uric acid, we are performing the reaction. So you have to put the reaction mixture at specific set point of temperature for a accurate measurement of reading of serum uric acid. So that particular instrument we can put in a incubator. Sorry, that particular reaction mixture we can put in an incubator. Okay. Now this is a water bath. Okay, this equipment has water filled in it and the temperature of which can be controlled by adjustment. This is used for various experiments where a specific temperature is used for heating the reaction mixture. Almost same principle of incubator and water bath. It is commonly used in medical undergraduate practicals where heating of solution is desired. In this equipment, simply sample can be heated at constant temperature for a prolonged period of time. Okay, right. Now this is pH meter, very easy instrument to identify. You would have to just identify this glass electrode. Okay, you have to identify this glass electrode. And here you can see the digital screen which shows a pH reading of a particular solution. Okay, you have to dip the glass electrode in a solution and it will measure the reading. Okay, this is not required at level of you, but whenever you want to see, you can see by zooming in. Okay, right. And this is also, you know, what is the pH of normal plasma, 7.35 to 7.45. This is important point. Urine ka 6.5 hai, CSF ka 7.32 and gastric juice ka 1 to 2. Okay, you know the normal principle how the pH is working. You should know the formula according to your chemistry practicals. Okay, I am not discussing it here in a detail. Okay, functioning principle you should know. Okay, now these are various glasswares. Okay, this is various glasswares. Okay, these are various glass instruments that we are measuring and we are using in a UG lab. Okay, this is thermometer to measure the temperature. This is graduated cylinder. Okay, whenever required we put the solution in it to measure the quantity. So this is a volumetric flask. This is Griffin beaker and this is Erlenmeyer's flask. Name you should note of this both of the two name is required. Okay, right. Now this is very important instrument. This instrument we are using in clinical chemistry lab. Okay, this is semi automated analyzer. Okay, and next instrument I am showing you, this is a fully automated analyzer. So what is the difference in semi and fully automated analyzer? So whenever you want to do any test or any biochemical test of any parameter, like say serum urea or serum uric acid, okay, or SGPT, SGOT in liver function test, Okay, we have two options. Either we will do from semi-automated analyzer or we can do in a fully automated analyzer. Okay, so in a semi-automated analyzer, I have to take the sample. Okay, serum sample I have to take after a centrifugation, and I will take that serum and I will mix the reagent for a desired substance to be measured. I will put on that particular reagent, and to this I will be adding the and I will be incubating the sample, and that will be giving the result, that semi-automated analyzer. You can see the sample aspiration probe. I have to put the sample here. It will aspirate the sample, and according to the colorimetric principle, this machine will show me the result. Okay, you can see the printer also. You can generate a printed report also. Okay, so semi-automatic biochemistry analyzer is capable of performing routine biochemistry tests. What are the routine biochemistry tests? We are performing the biochemistry lab. So first test is LFT that we are performing. LFT will include SGPT, SGOT, alkaline phosphatase. That is then so albumin, globulin, okay, total proteins, and serum bilirubin, direct, total, etc. Okay, so these are the LFT that we are performing. Then we are performing RFT also, renal function test. What are the renal function tests? Serum creatinine, serum urea, etc. Apart from it, some enzymes of cardiac biomarkers like CKMB, troponin I, okay, acute inflammatory markers like CRP, then serum ferritin. Okay, these all will be performing in the biochemistry lab. Okay, right? LDH, some drugs like lithium. Okay, lithium also in a antipsychotic patient or antimaniac patient. When the lithium is started, we have to measure the serum lithium levels to avoid the toxicity. That is also measured by our lab. Okay, so this particular instrument is used on photometry principle. Okay, semi-automated biochemistry analyzer is capable to perform tests on a whole blood, serum, plasma, cerebrospinal fluid as well as urine. In a small site laboratory, it is suitable. Okay, जिसका टर्न अराउंड ज़्यादा नहीं है, with this is small size of laboratory के लिए we can use that particular analyzer. Now this is fully automated analyzer. In clinical chemistry lab we have both the analyzer. Okay, fully working analyzer is CCL lab में you can see, you can visit the lab and you can see this particular analyzer. Okay, right, I will be explaining, I will show you the video of how the analyzer is working. Okay, but that for that we will do in a next video, not in this video. Okay, otherwise the video length will be longer. Okay, so you can see the reagent carousel in which the reagents are put. You can see the sample carousel. Okay, at the same time I can put so many samples, 50 to 60 samples I can put on a same time. Okay, and it will give the result according to turnaround time of that particular test that has to be done. Okay, so fully automated biochemical analyzer has automated sequences of operation which are used to be conducted manually in the past. Okay, and we are going to perform some test manually when we are starting our colorimetric practical. Okay, that colorimetric practical you can be asked in your university exam. All of you have must one practical of that colorimetry, that is of 35 marks. Okay, that is major exercise, right? So it is a colorimetric based principle, and fully automated analyzer is working according to colorimetric principles. Okay, this is how you can see the fully automated analyzer is working. We have reagent terminal, reagent turntable. There are two reagent turntable you can see in this photo. Okay, in this diagram, right? Okay, we have sampling pipette. They will collect the sample, and we have reagent pipette also that will collect the reagent, and there is a mixture chamber apart from it, and then they will be analyzing according to colorimetric principle. Okay, you can see this is a dispenser. Okay, very basic instrument, but yet sometime we put in a spotting examination. Many students fail to identify this. What is this? Okay, so it is a laboratory bottle dispenser, which is specialized tool designed to dispense a precise amount of liquids or chemicals when from a bottle to another container. So whenever it's dispensing reagents, solvents or acid, this equipment ensures that measures are accurate and precise. So it will prevent a potential error in your experiments as well as research. Okay. Okay, now little bit we are focusing on the blood collection system. I already told you we will be discussing this all things, okay, in a separate lecture of blood collection system and sample collection, okay. But to give or to sake of the completion, these things is going to be asked in your practical spotting examination. One question must be there from this particular thing. Okay, so this is called as vacuitainer. There are different types of vacuums, they are available. Okay, in the vacuum we are collecting the sample. Okay, and there are so many types of vacuum. This is a color is universal color. Okay, right, I will show you first. Yeah, see, this is a vacuums. Okay, and I will discuss this. Like suppose this black color one is a sodium citrate vacuum, in which citrate concentration is 1 is to 4. This blue or light blue color is also sodium citrate vacuum. Okay, but here the concentration is 1 is to 9, right? This red color tube is a plain vacuum. Okay, plain. This purple color is a EDTA vacuum. Okay, you can see the EDTA vacuum, ethylene diamine tetraacetate. This is gray color. Gray color vacuum is a fluoride vacuum. Okay, fluoride vacuum is of gray color. Green color one is a heparin vacuum. Okay, heparin vacuum. And this is a new one. This is called gel clot activator tube. Gel clot activator tube. Okay, this is yellow colored. This is same as whatever test to be done in plain vacuum, that all tests will be done on gel clot activator tube. What is the advantage of gel clot activator tube? I will be discussing shortly. Very basic thing I want to tell you. I have asked already the question serum and plasma. Okay, what is this? So when you put blood in a plain vacuum, we have not added any anticoagulant. Okay, when you put the blood in a red vacuum and you allowed it to the clot, all the clotting factor is utilized, right? And whatever the supernatant is there in a red vacuum is your serum. Okay, while we put a blood or whole blood in a EDTA vacuum, so EDTA is anticoagulant. Okay, so once a blood will be settled down, whatever the amount is left above is your plasma. Okay, right? So plasma, if I remove all the clotting factors from the plasma, it will lead to serum, right? This thing is very clear. Yes. Okay, now we'll discuss this vacuums one by one, and what are the different tests that has to be done in this particular vacuums. Okay, so whenever you want to do any biochemical test, you have to collect the blood in a specific vacuum. Okay, the color coding is very important. So now first we'll start. You can see there are two red colors vacuums are there. Okay, you can see two red color vacuums are there. Sorry, yeah, you can see the two red color vacuums are there. Okay, so first red color vacuum you can see, which is made up of glass. Okay, whenever the red vacuum of glass, the clotting time is 60 minutes. Okay, but the mixing instruction you should not, you do not invert it, the vacuum after collecting the blood. Purpose of this vacuum is you have to do, whenever you have to do a test that is to be done by the biochemistry lab, you have to send the sample in this particular vacuum. Okay, now we are not using the glass vacuum, we are using a plastic vacuum instead. That is also having a time of 60 minutes, but in the glass vacuum there is no additive, but in red vacuum we have a silicone coating of it. Okay, this is the difference. Okay, also purpose is serum biochemistry lab. Whatever the test done by the biochemistry, you are performing this particular test. Okay, like SGPT, SGOT, renal function test, liver function test, thyroid function test, all will be done by this particular biochemistry lab, and you have to perform, you have to take the sample in this particular vacuum. but the mixing instruction you should know, you have to invert it 4 times. Okay. Now this is yellow color. I have already shown gel clot activator tube. Okay. The clotting time here is drastically reduced by 30 minutes. You can see here the clotting time is only 30 minutes. So whenever you are performing or you are taking the blood and you are sending in a biochemistry lab in a clot activator tube, okay, your serum is separated in a 30 minutes, only 30 minutes. Okay. Also, you have to invert it in a 4 times. I will be showing you whenever I am taking the practical. Okay. I will show the photograph of the clot activator tube. Okay. Yeah. See, you can see this. This is a clot activator tube. Okay. You can see very nicely put at the bottom. This is a gel clot. Okay. Gel clot. Okay. It will take a 30 minutes to clot and to separate the serum. Okay. Okay. Now you can identify it. Okay. Yes. Okay, so this you should be keeping in the mind. We can put this vacuum and we can show the photograph of this vacuum when you are performing the examination and in the spotting examination. Okay, you all must know it, right? Right. Okay. Now you can see this orange color vacuum in which clotting time is 5 minutes. Only 5 minutes. Okay, this is called rapid serum tube in which your gel clot, whatever gel clot is there at bottom, is a thrombin based clot activator. Okay, this is a very newly formed vacuum. Sometimes it is used when the samples are, we want sample result at a lesser time. Okay, right? I will show you the vacuum. Okay, see this is a rapid serum tube. Okay, in which you can see this, the clot is there. This is a thrombin plus clot activator. Only in a 5 minutes it will separate a serum. So clotting time is 5 minutes here in this particular vacuum. Okay. ज़्यादा जल्दी रिजल्ट चाहिए होता है तब we can collect this, but it is costly. Okay, it is a costly vacuum, right? Now light blue color vacuum is sodium citrate vacuum. 1:9 ratio होता है. Coagulation studies में you can include a PT, APTT, INR. So whenever you are performing this test, you have to take the sample in light blue color vacuum and you have to invert it in a 4 times after collection. You can see the black color sodium citrate 1:4 ratio coagulation study. Apart from it, you can take this particular sodium citrate vacuum in for ESR measurement also, erythrocyte sedimentation rate. Now green color vacuum, you can see this is called heparin vacuum. It is used for arterial blood gas analysis and you have to invert it in 8 to 10 times. Okay, to mix the heparin and well with the sample. So you have to preserve that particular sample. In the arterial blood gas analysis, we are not taking the venous sample. We are taking arterial sample by arterial puncture. Okay. We will be discussing when you are discussing the arterial blood gas analysis. We have separate chapter for it. Okay. Then you can see the lavender color vacuum, that is EDTA vacuum, most common vacuum which is used in pathology lab. Okay. In a pathology lab we are using pathology lab mein EDTA vacuum is most commonly used. Okay. And in a EDTA you can perform a CBC, hemogram, complete hemogram, ESR also, and HbA1c also. Gray vacuum, you can see this potassium oxalate and sodium fluoride vacuum. Because why we need sodium fluoride in it? Because we want to measure a blood glucose, that is RBS, PP2BS, or fasting blood sugar. You should take the gray vacuum. You should take the blood in a gray vacuum. Why? Because fluoride will inhibit a enolase enzyme, which is a enzyme in a glycolysis step. So whenever I want to measure a glucose, I want to measure in a correct amount. Okay, you know, whenever the glycolysis process is happening, the glucose is going to be lysed. Okay, glucose is metabolized. I want to stop that particular metabolism because I want to measure exact amount of glucose. So in a blood glucose measurement, I will take a gray vacuum. One catch point is here. Okay, whenever there is a delay, you know, whenever there is a delay, okay, in sample transportation, you should use a gray vacuum. But you can do random blood sugar assessment in plain vacuum also whenever you are performing the test. Okay, there is whenever there is no sample delay, you can put the blood in red vacuum and you can measure the RBS also. Okay, I hope this thing is very clear. Vacuity part is also very important. Okay, You must know, you have to revise this practical. Okay, any doubt in this particular practical, you post in my comment section. Okay, right? Okay, I want to discuss this particular thing also. Okay, this is not going to be asked in your exam, but I want to give the extra age information to you. Okay, you can see both the acutes. One is a dark purple color, lavender color, and another one is a pink color, slightly violet, violet color. Okay, so this is your K3 EDTA acute. Okay, and this is your K2 EDTA acute. Okay, both are EDTA acute. Okay, both will be performing the similar test. Then why? What is the need of different vacuums of one name? Okay, so you should know K2 EDTA acute is slightly more advantageous than K3 EDTA acute. And why so? First point, K2 EDTA anticoagulation has no dilution effect. Okay, it has no dilution effect. Important point. Why it is important? So whenever there is no dilution, it will preserve accurate blood cell count and indices in it when you are measuring. Okay. Second advantage is whenever there is no dilution effect and all, you can see best or better morphology of RBC and WBC whenever you are measuring. Okay. Third important thing, according to some lab studies that have shown, K2 EDTA vacuum is more compatible. Okay, more compatible with automated analyzer. Okay, right? Okay, I hope this thing is very clear. Okay. Moreover, fourth advantage is it is a dry form of anticoagulant. So it will prevent a contamination. Okay. So for a longer period of time, you can store this vacuum without collecting the blood. Okay, this is very clear. I hope this thing is absolutely clear. This is just for your knowledge. No one is going to ask you this in an exam. And whenever it is asked, you are at the extra age information. Okay. I hope this practical is very clear. Any doubt you can post in my comment section or you can directly approach me. Okay. Thank you so much. We'll see you in the next practical class. Okay. Best of luck.